He won a Nobel Prize for describing structures of proteins, studying structures of proteins. And a lot of the work was done down off of Del Mar at what I like to call it PCC South, or they like to call it Caltech. but it's our South campus. So he did a lot of that at our South campus. And then if we take, this could be, this could even be parts of the same protein, could have these different bleeded sheets and whatever there. It's just starting to fold, right? And then this part over here may interact with this part over here and start to fold over, because maybe these are all hydrophobic and these are all hydrophobic, so it's going to fold over on itself, right? So then we get this large-scale three-dimensional folding. And I don't know. Isn't that pretty?
Very three-dimensional. Yeah,
it is, So we would often draw that, maybe something like this. Right there, right? And we create something that now has 3 dimensions. And that level of structure there, with 3 dimensions, is called tertiary. 3 with the superscript. And that is 3-dimensional. All proteins must at least have tertiary structure, because they have to be 3-dimensional. They have to be able to recognize and bind to each other. There are a few proteins, actually there are more than a few, there are a lot of proteins that are made from more than one polypeptide. A classic example is hemoglobin. Hemoglobin is a protein. It has 4 polypeptides. Each one of them has tertiary structure but is not functional. And it's not until you get all 4 of them together that would make it functional. And that level of structure would be called quaternary, which means 4, right? Quarter. Quaternary structure, and it's a 4 with a superscript. So this is quaternary structure. You guys probably can't see the thing. Sucks for you. This is multiple polypeptides. So we'll see a lot. Like I said, proteins have to have 3-dimensional structure to be functional. If it's one polypeptide that makes it functional, we say it has tertiary structure, and that's the protein. If it takes 2 or more polypeptides to be functional, that would then have quaternary structure. Each independent subunit would have 3-dimensional structure, but wouldn't be a protein. So proteins, the best definition for a protein, in 2 words, functional polypeptide. It can do something. So that's what we're looking at there. So if we look our way through these structures drawings, this is